We are doing an unfair fight tonight. I'm challenging Nathan with Topoisomerase evolution. How does Topoisomerase evolve? And Nathan's going to do his best to explain something that nobody can explain. So welcome, everybody. Welcome, Nathan.
It's so unfair. You're so unfair coming up with this topic. Sorry.
It's just one of the many chicken and egg problems for evolution. So let me just introduce really the problem here. So here we have, this is one version of topoisomerase. This is a protein that there's versions of topoisomerase in every form of life that we know. All plants, all bacteria, everything that has DNA must have topoisomerase. What topoisomerase does is it's like a little scissors, okay? Now, you have DNA, and whenever your cells need something, something is copied from your DNA, or when the cell divides, the DNA has to be copied. Now, when that happens, the DNA gets twisted up as if, you know, you're twisting a garden when you've got your garden hose, it'll get twisted up after you use it for a while. Well, the same thing happens with DNA. It gets coiled up, and what topoisomerase does is it comes in and it's like a little scissors. It snips the DNA, untwists it, and then splices it back together. Now, if that doesn't happen, the cell cannot transcribe, it cannot do its function. This is death. And in fact, topoisomerase is when there's tumor cells, this is one of the things that our cancer drugs targets, because if they can kill the topoisomerase, the cells die. So these are the challenges related to topoisomerase that I see. And by the way, these are several different versions of topoisomerase. So problems with topoisomerase evolution. Number one is a sequence problem.
Well, before you get into the problems of the evolution, I wanted to, just to let everybody know, as soon as Rebekah says what the topic is, I'm not allowed. This is on my honor or whatever as a Cub Scout or Boy Scout or whatever, but I'm not allowed to research this topic. And I want to let you know, I have absolutely, zero knowledge about this particular aspect of biology. So if we have any unfair friends, people who know something about biology, please throw me a lifeline. So a lot of what I'm going to be talking about tonight is all, hypotheticals that I've come up with knowing almost absolutely nothing about this topic. So if you have any information or can help me in any way, that would be great. Like, I didn't even know there were different versions of topoisomerase. So I'm just trying to kind of slow it down and assimilate this. But generally speaking, as I understand it, the chicken and the egg problem here, and this is what I've been thinking about, is that in cells as they exist now, you require these proteins to kind of do some on the fly organization of the DNA before the cell can replicate. Is that correct, Rebekah? So you have the G1 phase, the G2 phase. Between when it's at a G1 phase and it's moving towards the G2 phase, and it's in the S phase, I think it's called, of cell division. Is that when topoisomerase is organizing and multiplying the DNA? Is that right?
Remember, it's not multiplying DNA, okay? When the DNA is being transcribed, the DNA gets twisted up, okay? And if it doesn't get untwisted, then the transcription isn't going to work. So the DNA is getting twisted up. Topoisomerase comes in, snips the DNA, uncoils it, and splices it back together.
Okay, so it's in protein synthesis. It's before, it's as it's about to create some sort of RNA molecule that's going to go out of the nucleus. Is that right?
Well, it's okay. There's different versions of topoisomerase, and they're used at different times. And I'm not an
Okay.
expert on all the different types. In humans, we have 6 different types. Okay. So we have
Okay.
6 different
Okay.
types of topoisomerase, and they appear and do the thing whenever it's needed. Okay. But there's this, I can't go into the weeds on which topoisomerase is needed at exactly which time.
Okay. All right. So we have this toolkit, let's say, of proteins that are used in the synthesis, in the processes of the cell to generate other proteins, or to do whatever, right?
Yeah, and topoisomerase is just one of these. Okay, so we could probably pick 1,000 and chicken and egg problems just inside a cell. Okay, but
Okay.
today it's topoisomerase. So
Okay. Topoisomerase, and hopefully we don't have 1,000 unfair fights about this. So I'm going to try and get at some of the philosophical things behind this and see if we can approach it that way.
okay, well, and by the way, I've already chosen the next challenge I'm going to do for you, and I picked it both on what would be easy for me, but also what would be fun for you. So I'll
Okay.
just give you a clue. We're going to space, so you'll be
Okay.
happier with that.
Finally.
Okay, so I won't keep you
Okay.
deep in the weeds, but I want the audience to know that I usually pick my topics based on there being no evolutionary answer. Okay, so I only pick topics that it really doesn't matter if Nathan has a chance to research it or not, because he's not going to find anything that's going to help him here. Okay,
Well,
so
and I also enjoy this. I mean, I enjoy the theory crafting, the hypothesis building, because it makes me, like a lot of my thinking leading up to this is about, biosynthesis. How do you get life from nothing? But it's like very kind of first principle. So it is. I do appreciate that we have a little bit of information about this particular thing. Anyway, please continue with the details. What are all the problems with this, with evolution?
okay, yes, here are the problems. Now, number 1 is a sequencing problem, because topoisomerase, the one that I'm showing you right here, it's comprised of 1,531 amino acids in a particular order. Okay, so in order to get this fantastic little machine, you need 1,500 amino acids to line up in the right order. And you also, that brings us to the second problem, the problem of protein folding, which is it has to know how to fold itself properly. And we don't even fully understand how that works. Okay. Number 3 problem, the chicken and egg problem of topoisomerase, because how do we get topoisomerase? It's coded in the DNA, so it has to be transcribed in order to be built. That DNA section has to be transcribed in order for the topoisomerase to be built. So, but the problem is you've already got to have topoisomerase or you're not going to be able to build a topoisomerase. So it's a classic chicken or egg where you've got to already have it, but you've also got to build it. So which came first, the DNA to code for it or the actual thing itself? Now, the fourth thing is that topoisomerase, this requires foresight, right? There has to be something that has the foresight within the cell to know that the DNA needs to be untwisted. All right. And then number 5, that knowledge has to get to topoisomerase. How does a string of amino acids that's folded together, how does it know what it needs to do? It's a fantastically built machine, but how does the machine get the information that it needs to go over at this particular time and cut the DNA and untwist it and splice it back together. So it requires knowledge. Number 6 is that if, I was reading this paper on the origin and evolution of topoisomerase. Well, the problem is that the different types of topoisomerase are so structurally and sequentially different that they had to have multiple independent origins. And possibly the problem with that also is that it contradicts the tree of life. Okay, we cannot, and this is straight up in this paper, I've got a quote here that says, and I'll show it later if I need to. Actually, I'll just show it right now. But basically, it says the phylogenetic distribution of DNA topoisomerases is thus quite puzzling and clearly does not agree with the classical universal tree of life, neither with any of the alternative models, such as the bacteria first model, nor with the ring of life model. So we cannot get topoisomerase to fit in this evolutionary tree. All right, so now with that, Nathan, you're free to make your explanation for how topoisomerase came about.
Well, thank you. You were talking about, you showed that picture, and it's actually many, there's a class of proteins that are performing certain functions, and maybe they're all called topoisomerase or topoisomerase or whatever, however, topoisomerase, let's just say it that way. And so they all seem to do some sort of function regarded to manipulation of DNA. And so, thinking through this, I thought in my mind a little square. On the upper left, we have creation, right? Like all those things you said, it has to have foresight. It has to have, some sort of, God knowing the whole plan and then manufacturing the pieces and putting them together, essentially, right? And then, on the top right, we could think of DNA first. The DNA existed first, and then topoisomerase, and this is the chicken and egg. Oh well, you have to have topoisomerase for DNA, all right? And then in the lower left, we have the opposite, topoisomerase coming first. But how did it come about without DNA, right? And then, but there's another square that that I think is interesting that people might not think about, and that is that neither of them came first. And I think that this is, I think at some level, all of these are, true, but it's a matter of timing. And so, when I was trying to think about how would these things come about, I think that there's kind of a hierarchy. Maybe we need to go into this on another topic, but this is just kind of, my weird thinking. Okay, number one, I think that you have to have geometry, right? So you have some sort of structure before there's life, right? Because the other thing that you said in the topic is that topoisomerase is in every cell, right? And I think that was a bit misleading because you said, because then I was thinking about this particular topoisomerase. But what it sounds like is that topoisomerase that you showed me is not in every cell. There is some sort of protein that functions to manipulate DNA in every cell, right? And so if I go back down this tree of life, then it does bring me back to before there was DNA, right? And so you have to have some sort of mechanism to get DNA, or the start of DNA, and you have to get something that's starting to manipulate it. And I think that goes back to the very beginning. And so with topoisomerase, so let's just say that it's something that rotates DNA, right? I think number one, the DNA we're talking about is super, super, super long, right? We have kilobase pairs, and I don't even know how big the whole genome is, right? And so you're talking about something very long, like you have a very long garden hose. But if we think about the origins of DNA back towards the beginning, it probably wasn't that long, right? And so would it need to be manipulated in all these complex ways? And I would say no, that the length of DNA extended over time, and so the number of tools that were required to manipulate that DNA increased over time. So we're talking about an increase of complexity over time. And so I think that the tools that began to manipulate DNA may have had a different origin than DNA, right? And then over time, both things evolved. The DNA got longer, and the tools that then were required to manipulate it became more complicated, and a wider variety of tool sets. But the fact that it's a basic geometry thing might indicate why this circle of life or ring of life or these things, tree of life, whatever the explosion of life, whatever you want to say, to talk about this overall panorama of it has changed. I don't know. Is that just gobbledygook? Do I need to go more deeply into? I don't want to just mess people up with philosophy. Uh, Go ahead.
Well, I mean, I think you're doing as best you can under the circumstances. By the way, I have to give a shout out to Sal, because Sal's the one who taught me about topoisomerase. Good creation, buddy, and really made me learn this about this protein. So thank you, Sal. Okay. Well, I think you're bringing up an interesting point about DNA, but I will say this. We have tried to figure out what could be the simplest organism that could survive, right? And one thing that we've done is tried to basically knock a bunch of genes out of a bacteria and see how many it can survive without. And so they were able to get a pretty crippled bacterium, and by knocking out a bunch of genes, but they realized they had to at least have 480 genes for the creature to live. And so 480 genes translates into, I don't know how many millions of base pairs, but genes are usually thousands of sequences long. So, I mean, even what you might consider short DNA, it's hard to imagine a cell or a functional organism that would require DNA that would be short enough that it wouldn't need this function. But I mean, even if there was, it seems a little bit strange that then the DNA would evolve a problem that would then need to be solved, right? Like, what I mean is, and then you've got the problem of you've got every form of life that we know today. So this would have to be going back to the, Luca or whatever, the last common ancestor. And so it's unclear that we could have this situation. It would seem a little bit strange that we wouldn't find anything that doesn't have this functional protein, and it's being named by the function that it produces. So you said it was misleading because I showed this one, but it's being named by the function. So right. Okay.
And yeah, so let me address that first. I don't believe that there is a Luca. Like when you're talking about them trying to synthesize the simplest organism, when you're trying to synthesize a cell that can replicate and do all these functions, I think you are already at a level of complexity where something could be, semi-autonomous, you know what I mean, with its environment. And I think that the origin of life goes obviously back much further than that. And that it is not a single common ancestor, but it was a system. And part of the problem here is a problem of knowledge, right? So when we look at chicken and egg, if we get away from proteins and just talk about chicken and egg, which came first, well, a person who believes in evolution and that the Earth is old believes that there were millions of iterations leading up to chicken and egg, right? And so the problem we have is a problem of knowledge. Like, if I said, okay, there was a state of the world in 1901 at 12 a.m. that led to the present day, we could make probably a pretty good guess that that was true, right? But if we think about what was the exact state of the Earth in 1901 at 12 a.m. that led to now, it would be impossible to know what that state was completely, right? So there's a problem of knowing. So the closest I can get is start to approach things as in what ways is it useful to think about this in order to arrive at the current solution so many iterations later. And for me, like I said, I think it's actually systems, not one particular common ancestor. And what do I mean by that? Well, when I was thinking about this again, because all I've had to do is think about something I have no knowledge of, I thought, okay, I think that it has to start with geometry. Okay, and what kind of geometry? I think some sort of almost perfect tessellation. Okay, so tessellation is like a mathematic thing where you can put these shapes around and they fit, but it never replicates, right? So some sort of tessellation that's almost a perfect, able to completely tessellate. And I think before there was DNA, the oldest part of a cell is probably something more close to a membrane. Right, so you have this replicating pattern of proteins that form this kind of shell, but it also has kind of irregularities along it where these proteins stick out. And I suspect, like if we took that model and we said, well, what's something that's similar to that, but that is purely chemical? I think of soot. When you have these kind of this simple geometric pattern, which is these carbon atoms in a hexagon, and you put them all together, you get these irregularities in it, and it forms this kind of sphere type thing with multiple layers going round and round and round, right? And the perfect one would be a buckyball, right, at carbon 60. Sorry, this is where like I'm getting really deep into this, but I think this goes way back, right? Because you're asking me something that goes way back. And so I think that the next step after geometric is physical topographies, right? So you have something that's kind of a creating a semi-spheroid, but has holes in it, and that's creating charge differences, pressure differences, and concentration differences. And so if I thought about what was the start going towards life, I think you would have a bunch of these things that are all different. And some of them might have this charge disparity where something comes in, gets flipped around, and goes out. So you just have this kind of, it's attracted by the positive side, goes in, and it's already got an acceleration. Like, have you ever taken a straw and dipped it in water, right? And you brought it out and you have the fluid stuck in there, right? You've done that? Okay. Now, if you take that straw and you put it, we're pretty far from Topoisomerase, but if you put that straw back in the water and let off your finger, you'll notice that the water equalizes, but before it does, the water actually gets higher than the level of the water and then drops back down. Have you noticed this effect? Yes.
Yes.
Okay. So in the same way, I'm imagining the beginning of things, right? And we've got all these, things floating there that are, it's just chemical. But there might be, among that myriad of time and everything, something where you have a charge on one end and you have a charge on the other that's opposite, and something can float in and it's got an acceleration, but as soon as it's in there, the polarity changes and it has to flip, but it because it has an acceleration, it doesn't match the polarity just correctly, and then it floats out, right? And you kind of have this. Well, the first topoisomerase that I can imagine, the proto topoisomerase, is something that's stuck on the inside of that wall. It's something that has a slight charge of its own, and it's kind of stuck, and so it's kind of like a jagged edge sticking out of whatever surface that's creating these differences in topography. And so when that thing comes in, instead of flipping randomly up toward the top, it now has an inclination to flip a certain direction. Let's say it spirals once and then goes out. Spirals once. And to me, the thing that's coming in and going out is kind of a very simplistic toy model, whatever, of DNA. And polysomerase is this thing sticking outside. So there you have it. So if you go back millions and millions and millions and millions of iterations, right, then that simple thing of muck sitting in the ocean of, whatever, is kind of the beginning of these two in terms of function, right? Um, so that's kind of my idea. So you have physical topographies, and you have some sort of transport that occurs, and I believe that there are multi-system synergies. So, for instance, this whole thing, this toy model I've created with this thing, that's not enough to get you to the first cell. You have millions and millions of these things, and they're all kind of doing something different. And maybe there ends up being some synergy between one and another, and this one spits out something, and this one pulls it in, and you get the next iteration, right? But there were billions of years before, at least as I understand it, billions of years before life popped up on Earth, and so
All
you have to
right.
have,
Look,
yeah.
the chat's going crazy. They have a lot to say to you, Nathan.
All
I
right.
have some unfair friends here tonight, so
Okay.
I'm glad they're here. But I wanted to, and I'm going to put up their comments in a minute. But I just want to say, like, wow, you have a good imagination, but you're a long way from getting life. But this
Yes.
isn't about the
Yes.
origin of
Because
life.
you're saying you're giving me the finished product. You're saying how
I'm
do you
giving
have
you
a Toyota here?
I'm giving you one thing
Yes.
within about a billion part
Yes.
cell. Okay,
Yes.
one out of
Exactly.
a bill. I'm giving you one
Exactly.
part at.
That's right. And you're
So,
saying, okay, here's this
but
billion
but
component
we're not
thing.
doing
Okay,
the
tell
whole.
me.
We're not doing the whole origin of life. And I have to make this point before I put the comments up, because you went on such a long tirade, but I have to make this point. You said the chicken and the egg problem is really a problem about like missing knowledge. No, the problem is that you need both simultaneously. That is what the chicken and the egg problem is. It's that you have to have simultaneously a chicken, and the chicken has to have the ability to lay an egg, and there has to be a rooster, or you don't get the next generation. You don't have millions of years to evolve by a step-by-step process. And that is true for many things within a single cell. And we're talking about one of them tonight. You don't have millions of years to develop this over time. This is something that has to be there, or there's death. And it's not a lack of knowledge problem. It's a problem of needing simultaneous things to happen. So that
Well, I want to see the comments. Let's get the comments.
All right. Yes,
My
let's
long
go to the
tirade. Sorry.
Now, this one is sort of against me, but Otangela says, hi, Rebekah, the origin of topoisomerase is an origin of life problem, not evolution, since it is life essential protein. Well, Otangelo, you're right. I usually hear this in origin of life, but first of all, most people want to push it away from the origin of life and try to make it that this came later. As you notice, Nathan did that at first. He said, I imagined that the first life was more like some DNA and blah, blah. So they want to push it. So whether it's origin of life or evolution depends on where someone thinks topoisomerase originated. Yes.
Thank you so much for that comment. And isn't it interesting that we independently arrived at that solution, that it's actually an origin of life problem?
Okay.
Wow.
Well,
two parallel evolutions of thought come together and spash, knock you in the face, Rebekah. Boom, boom, boom. Independent, co-evolving thoughts coming together and smashing you, like topoisomerase and DNA coming together from separate,
It's only my merciful heart that allows people to separate origin of life from evolution. This is a mercy toward the evolutionists because they're so desperate to get origin of life away from them. They want to make it a separate thing, but it all actually goes together because we're talking about material processes being able to produce life and the diversity of life that we see. So it actually does all go together. But since the evolutionists are so desperate to get origin of life problem away from them, then I usually let this go and let it be a separate problem.
Okay. But just the fact that there's a 2 under your, on your slide, I'm just, keep in mind, I'm going from the bare essentials. All I have to live on for topoisomerase is what you've given me. But you've given me insight by putting 2 there, topoisomerase 2. So if we were really talking about evolution here, then we would just be looking at variations in topoisomerase among different species, right?
Okay, wait, maybe you're confused about what the 2 is because I didn't really explain. The topoisomerase 1 cuts 1 strand of the DNA to relax the DNA. The topoisomerase 2 cuts both strands of the DNA. So I chose that one because I think it looks cool. It had the coolest picture, but I mean, I don't think that
it doesn't matter. The 2 doesn't matter.
Okay, right.
Yeah. Okay.
Okay, so let's go to the next comment. So anyway, Otangelo says phospholipids are very sensitive to perturbation and disintegrate with ease. So he's coming against your scenario that you were describing. He says you need calcium concentration 10,000 times less inside the cell than outside to get homeostasis. You need membrane proteins. How do you get them prebiotically? Then,
okay. Wait. Let me address that.
Okay, there's a lot more. There's a lot more.
Okay. So, real quick with the membrane. I'm not saying that it was a cell membrane. I think that again, the modern cell membrane of a cell is like a Porsche. Okay, and I'm talking about a stick. At the beginning. So what I'm saying is that it wasn't anything like a cell. It didn't look anything like a modern cell. But what you need is some sort of separation. You need some sort of physical topography that separates one thing from another to start to get these types of differences that you see now. Like you said, in a cell, it's tens of thousands. It's 5 orders of magnitude different. But I'm not saying at the beginning it was 5 orders of magnitude different. I'm saying it was 0.001% different, right? It was 8 magnitudes, whatever. It was almost the exact same as what was outside, but you had some sort of start through a charge of something that's happening. And we already see that with chemicals now. I mean, you have charges and, so I think that what I'm talking about with physical chemistry is present without any type of biology,
Okay, but let it be known that the chemistry required for life is not something that happens naturally. That's not something that wants to happen. But anyway, next comment, pigs can fly. Matt, good to see you. I haven't seen you around in a while. And I'm glad you're here, and I'd love to have you on the channel sometime. But he says, does he not recognize the information problem with this argument? So where is the information for building topoisomerase?
Yeah. Okay. Well, I do think that information is important, and I do think that in my abiogenesis, there is something about information there. I think when we think of information, we are already thinking about a fully developed cell, where you have replication, right, occurring. So this is information that replicates itself. But I think, if we think about information in terms of entropy, then I do think that even in my example, you have to have, some sort of external, light source, right, like the sun or radioactive decay or whatever, and that, ultimately life is a more efficient process from converting light or energy that's useful to light that is less useful for work. And so, but just like that straw where I drop it down and it comes back up momentarily before it drops again, you could think of life as kind of like that. It's kind of like it's a state where it's useful, um, in an intermediate step, kind of between the heat death of the universe or whatever and the Big Bang, in order to get to heat death faster. So.
Okay, well, howdy to Hillary's emails. Jamie says, God bless, hit like and subscribe. All the cool kids are doing it. Thank you, Jamie. And Otangela says, gobbledygook, Nathan. Without encoded information, you'll never get topoisomerase.
Okay. All right.
Matt says, Nathan, are you aware of the proton gradient that is necessary to keep the cell from destabilizing and killing itself?
Yes. Look, I don't know the exact specifics, but I do understand that if you stick a cell inside of distilled water, it's going to like explode or something. I mean, but all of these problems that people are raising, you already have a complete cell. And it's amazing. I'm not saying that I'm gonna just mix a bunch of crap together on my table, assuming I was in a clean room. I mean, even NASA can't keep life out of their clean rooms, so it's going to be totally compromised anyway. But I'm not saying that in one day I'm just going to throw some things together and suddenly topoisomerase is going to come out and a cell membrane is going to come out and all this stuff. I'm just saying, when I was thinking about what the most primitive proof of concept, if you take the function of topoisomerase, if you take the function of that and you try and distill it down to its most basic component is the ability to spin something or to turn something or to, interrupt something. And I am not going to be able to, especially with my lack of knowledge about this particular protein, even take you from N to N-1. If you think about all the iterations of topoisomerase, I can't go even from, I don't even know what N is. So how can I go from N to N-1 without a lot of knowledge about this protein? I can't.
Okay, well, you and the rest of the scientific community, because there is no answer to this, no matter how much information you have. The more information you have, the harder it is to get the answer for this. So there's a lot more comments here. Okay, so topoisomerase is info. It's encoded in the molecule. What was the mechanism before topoisomerase, Nathan? And so,
well, I already said it. I think it's as simple as something is in motion, and there's a charge, and it turns. It's not DNA, and it's not topoisomerase at that point. It's just a molecule, a few molecules long. But I think that that is essentially the repetition of that is, you have to start evolution somewhere, right? And like I said, in terms of the length of time of our planet compared to what I think our planet is, compared to how long life has been here, it took a long time to get to the most basic thing.
Well, and see, Nathan, that was a very telling statement. You said evolution has to begin somewhere, but that isn't true. Evolution does not
No,
have to begin somewhere. There could be another alternative. God created, right? That is an alternative that makes a lot more sense according to what we know about life.
well, but we know life is evolving now. You've even said that it changes over time.
It changes, but
Yes, okay,
within
okay, change over
what's
time.
all,
It's evolving.
well,
And
if that's
so
the way that
and
you wanted,
so if it's evolving now, it evolved in the past, right?
Nathan, you're using evolve in a very slippery way. I believe that organisms change mostly by what's already present, the information that's already present in the genome, the capabilities that are already established. Now, there's an additional way they can change, which is mostly harmful, which is by mutation. So that can also change an organism, and it's usually not a good thing. And it's definitely not a thing that builds new organs, new features, new systems in an organism. So, no, I don't accept this idea that just because, we might see a finch beak that's slightly smaller or larger than the generation prior, that means you can build topoisomerase and everything else you need for life by a step-by-step process.
Okay. So I'm going to take this, so we've gone to the origin of life stuff, and people obviously don't like that.
Well,
So let's talk about how topoisomerase could change now, right? And I think that one of the things that is problematic is for people to say, if the protein changes and it no longer functions the way that it does now, which I believe is true. I think that you do have to have all of these different proteins in a certain way, and if you change one, it's gonna change the folding and it's gonna change its function. But that's just it. I think it's gonna change its function, right? But if I look at all those topoisomerase, just briefly, the half second I saw that slide, I think most of them look pretty symmetrical. Is that right? They look symmetrical.
you mean the variations of topoisomerase? Let me just
Right.
let me pull up some of the pictures then.
Okay. So okay. So some of them are not symmetrical at all. So there that goes. What is this one in the lower left there?
Topoisomerase 2A.
And it's got different bits.
Yeah.
Okay.
So,
Well,
okay.
so I think one thing that's interesting to me is that you could have, and again, like I said, generations and generations, right? I mean, maybe I'm just inserting random gobbledygook evolution here, but I do think that we have to look at n and then n-1, n-2, and that kind of thing. And I could also see n+1, which is if you made one change, right? If one protein changed on this, could you show an efficiency change that degrades it, but it's still useful? It's just not as good as the current edition. Or can you make it better? And if you can do that by either removing one protein or another, that's one possibility for evolution. The other possibility is that if you change one protein,
do you mean
it
amino acid? I'm sorry.
amino acid. One amino acid. Or if you change one amino acid, does it stop doing this? And I think there would be multiple that would have to happen here, but would it stop doing this, and would it be useful for something else, right? So I do think that it's a multi-system process, and it's not just useful for the cell, but it could also be useful for viruses and things like that. So is there something about this where it could change based upon the needs of a virus, right? That it was partially encoded in a virus. So, looking at this, if I say from the evolution perspective, I went from the Genesis uh, Genesis part, but if you look at it from the end point, going backwards or moving forwards in evolution, then is there some way? And again, this is where you would have to have a lot of knowledge and experimentation to see where this could either improve or still work but be less useful by changing one amino acid.
Okay, well, that's something I can't answer. But let's look at these things and see which ones you've dealt with. Do you feel like you've dealt with the sequence problem that we need 1500 for this particular one I'm showing you, 1531 amino acids to line up? Maybe you could live with one or two of them. I don't know. That's something I don't know. But how would you get 1531 amino acids to line up properly for making this machine?
Well, it's not by snapping my fingers, I'll tell you that. I think it's a long process. And it probably went backwards and forwards many times. And so trying to trace the root of how this thing actually got to where it is again a knowledge problem.
Okay, well, I think it's more than that. But now, have you talked about how it would be known that DNA would need the uncoiling?
Well, again, I go back to length. So at some point, DNA does need these mechanisms to help it. I mean, it's part of all the machine. It's part of the machinery for how DNA functions, right? At the modern DNA, and so there was either a precursor to DNA itself that was functioning on some level, a single strand connected to something else, or I again, it's unknowable to me now what the precursor was, so I'm only speculating. But I do think that um, in its modern iteration, you need these things, these tools, all these tools in this kit. Like if I was assembling a car, I'd need a lot of tools and I need a lot of pieces. And the same is true with the cell. But that doesn't necessarily mean that there isn't something that's just slightly different that has all the same tools that is a little bit different, and then you go back a little bit, it's slightly different. I mean, that's what evolution is saying, right?
Okay, but in the meantime, then what do you do while you don't have the step by step? You know what I mean? Like you don't have time for the step by step.
Well, that's
So
that's what I was saying with the 1901. If you can't know all of it, then but you can still like come up with mathematical theorems in math that are useful for manipulating the information that you do have. And the same is true with evolution. Although we cannot, it's probably impossible for us to have all of the information regarding all of the states that led up to this current state. That's much harder than actually knowing the world at any particular moment, I think, in 1901. Um, so I don't think we're ever going to have all that information. So instead, we have to come up with systems and thoughts that allow us, that are useful for manipulating the information we have. And if you look at biology and what evolution, I think it is useful for manipulating the information we have.
okay, well, I disagree with you, of course, but so did you talk about how this protein can have the knowledge of what needs to be done? Now it has, we're assuming it was able to sequence itself, fold itself, and be present when it is needed, but how does it know about the job it needs to do?
Well, this is, I don't think it does know. I don't think it has a mind.
So is it just wildly running around snipping and it just happens to get at the right place? I mean, what if it doesn't know, then how is it done?
I don't, I think it's a physical process. I mean, do you not think it's a physical process?
It's a physical process, but what
Yeah.
I'm saying is there has to be knowledge somewhere. Okay. And it may not have the knowledge, but there has to be a mind that knows that that was what is needed and to be able to direct it to the correct location. So that may happen within the cell. It may happen. I don't, it may be just an automatic process that God set up. I don't know. But somehow it knows what needs to be done, or there is knowledge somewhere about what needs to be done. It goes to the right place. It doesn't just
I mean, I
wildly
think it's,
go sniffing around.
well, I think it bounces and the charges from something else bounce it somewhere else and bounces somewhere else, and eventually it bounces into this place where it's. I mean, you don't think this protein is floating around? You know what like what I'm saying is, I don't think it's completely chaotic. It's not chaotic, but I think that its individual emotions are chaotic. The Brownian motions of the different things are chaotic. Um, you know what I mean? That
You can speculate that it was like that in the past,
no,
but
no. I don't speculate. I don't think it was like it is now in the past, right? I don't think that when it was a soup, Earth was a soup, that you had this fully formed complex amino acid present. I don't believe that, right? I don't have to believe that to believe in evolution, right? So you're saying, how did this come about? And I'm telling you an incremental process, and I don't have the information to tell you what the step before that led to this particular one was.
okay. But I just want to make it known you're accepting by faith that there was a process by which this evolved. This is not there's no this is actually not according to our scientific observations. So it is a religious belief that this evolved by a
I,
step-by-step process.
no, because like I said, I don't have to have all the information to create a model for how the world functions. I don't have to know where every single star is and every black hole is in order to come up with ideas about their formation and life and death. But I can use the information I have to try and construct a story that makes sense. And I do think that evolution makes sense. We may not have the particular information related to this, but we might. I don't know. Um,
Well,
because
see, I don't.
no individual human has all the information either.
Well, I'll put it together and say, no, it doesn't make sense. And that brings me to this final thing that this actually contradicts the tree of life. If evolution is this process, we all evolved from a common ancestor and the tree of life branched out. Why can't we group it in one way that actually makes sense? If you look genetically, then you have to group creatures in a different tree of life than if you do it by morphology. And if we do it by proteins, we've got a completely different tree of life. So if there really was a common ancestor that we evolved from, why isn't there a consistent tree of life that makes sense with all the facets of what we see in life? I think
Well,
Nathan might be
well,
frozen.
I mean, when we say tree of life and common ancestor and everything, um, I think it's true on a macro level, but I don't believe it's true on a micro level. What do I mean by that? I mean, um, it goes back to the soup. I imagine one thing doing this wiggly wig, and another thing doing that wiggly wig, and you have billions of these things doing different wiggly wigs, and out of that, you have something emerging, I'm talking vague terms, but essentially what I'm saying is that things as a system were evolving, not as an individual organism evolving. And we talked about this a little bit, with the viruses and everything, that we are sharing information with other species all the time. And so it's not, so the tree of life is a simplification that might be useful, but it is like a web. It is like multiple things interacting, you know? And so it's a system, and then there's subsystems, and then you arrive at what we call an individual organism. But what is an individual organism even? Because if I look at you, Rebekah, if I put you inside of a machine and found out where all the information is, most of it is bacterial information, right? You've got billions of bacterias and stuff, of different things on your skin and on your hair and in your gut. I mean, so we're talking, even you as an individual organism. What does that mean, individual organism?
Well, that's going a little far off our topic, okay? But you finally brought up the viruses. Because for once, I was like waiting for you to say the viruses, because actually that is what the proposal is at the end of this origin and evolution of DNA
Well,
topoisomerase.
great. I mean, I did say, yeah.
Now they
and did you find a rebuttal?
That there's even a problem with that, and it's a very complicated problem, and we've been going a long time, so I'm not going to go into it.
Okay.
But they do suggest that it may have something to do with viruses and things like that. So it's interesting, because I when I read that, I was like, oh, Nathan's pet theory. Well,
well, let's say the problem we have is, like, let's say we're at in 15 million, right, for the iteration of this thing, right? And let's say 15 million minus 3 is when the virus happened. Then we still have to come up with -2 and -1 before we're at n. So I think it's like super difficult to really unpack and tease apart how exactly this went from this to just something slightly different, to just something slightly different, to something just slightly different, and then something maybe more major happened, but it was still very similar to what it is now. You know what I mean? It's just, I do think it's an information problem. And even if I did that for this one protein, you could just pick one of the other 1,500 ones and point at that one, and I would have to do the same thing for that. And so I do think it is an information problem.
Well, thank you for recognizing that, Nathan. And I hope we don't have to go through all 1,500 to show you so many problems, chicken and egg problems with evolution. But now I want to ask the audience, who won this debate? Don't be shy. Put it in there. And meanwhile, while we're waiting for the audience to answer, then Nathan, why don't you introduce the topic for next week? What's my challenge for next week?
Go Young Earth, V.S. Lewis.
It's a joke. It's a joke. Okay.
All right. Well, so Howard F., thank you. I think it was it last week? He said, do this topic, and so there's something called a paleosol, and I had never heard of this. And he said paleosols and river valleys in the geologic. What is it called? Geologic column. And so, I've just begun to research this. So obviously I'm not an expert. So if any of you guys know anything about this, or if Howard F. wants to get on and give some commentary, I think the issue here is that we have records of these different soils, and we have modern soils, that clearly have analogs. And there's a process ongoing with this soil. Soil doesn't just like pop out of nothing. It's really difficult to create in its current state, or difficult to evolve. It takes a long time for it to evolve in its current state. Because I saw Rebekah say, "Oh, he used design."
Well, I'm thinking
to
It does
create.
just pop out of nothing if God creates.
Okay. Anyway,
Okay.
so we're going to be doing these ancient soils, and how does the young Earth creationist who only believes the Earth is 7,000 years old end up with these columns that seem to indicate that there was an evolution of the terrain over time.
I'm looking forward to this one. And I'm not saying I have the answer for it. I don't, but I'm eager to ask a lot of questions. So try to know your stuff, Nathan. B.S. Lewis says, Jesus won.
Well, amen. Hallelujah.
Amen. Hillary's email says, Nathan gets my provisional vote. Wow.
Oh, thank you.
And then
Wow.
Hillary's email said, we all won from learning new things.
Thank
So,
you.
okay,
Yes.
and that's all the votes. Oh, D'Angelo, where's my vote? Just kidding. Okay. Well, anyway, guys, thanks for watching, and we'll see you next week.